Hello, girls and boys. Video number 2 is talking about the histology of the cortex, focusing on the renal corpuscle or Malpighian corpuscle. Look like yellow balloons on the models. Okay, this is the histology of the cortex, and this is the cortical lobule with the black arrow. Remember, there are no lobules in medulla. The border of the cortical lobule is interlobular artery 1, interlobular artery 2, sitting in the labyrinth. The center of the cortical lobule is called the medullary ray, where the straight tubules and ducts are located. You can see the straight tubules right here. Okay, what are the structures in these two locations? The cortical labyrinth, which is pink on the, labeled with number 1, pink on the microscopic slide, has the convoluted tubules. And these tubules look circles on the microscopic image. This is the 4 structures we're going to analyze in number 1, or cortical labyrinth. Renal corpuscle, pars convoluta of the proximal tubule, macula densa of the distal tubule, pars convoluta of the distal tubule. The medullary ray, which is purple and labeled with 2 on the microscopic slide, is the territory of the straight tubules and ducts. So therefore, they look straight. 3 structures: pars recta of proximal tubule, pars recta of distal tubule, cortical collecting duct. Remember, recta means straight. Renal corpuscle or Malpighian corpuscle, major function filtration on the model, little yellow balloons in the cortex of the kidney. Then magnified in one lobe, the yellow balloons, and one yellow balloon is magnified on the right side. Okay, I wanted to talk to you about the terminology. Renal corpuscle or Malpighian corpuscle is a terminology that combines the urinary system, where the urine is, that we call glomerular capsule, and combines the cardiovascular system, where the blood sits in the glomerular capillary. On this image, I am focusing only on the urinary side. We're gonna do blood vessels later. Number 1 is these purple round podocytes that has filtration slits are making the visceral epithelium or visceral layer of the Bowman's capsule. Number 2, you can see the flat nuclei all around. This is the simple squamous epithelium of the parietal layer or epithelium of the Bowman's capsule. So this is the visceral and this is the parietal layer. What's between? The lumen of the Bowman's capsule or glomerular capsule. This is where the filtrate or first urine will show up. Let's look at the same thing on the model. The yellow guys are the podocytes. The yellow is the cytoplasma, nucleus is blue, and these are the feet of the podocytes. Podocytes means cells with feet. You can see the little feet, and between 2 podocytes, the feet are connected with each other like puzzle pieces, and between the feet are the red filtration slits. Why is it red? Because you can see through into the blood. Yeah, there are podocytes on the right side too, but they left it out. Number 2, simple squamous epithelium of the parietal layer of the Bowman's capsule or glomerular capsule. And between the visceral layer and the parietal layer, here we go again, the lumen of the Bowman's capsule or glomerular capsule. Where does the urine go from here? Into the proximal convoluted tubule. You can see it on the model, but you cannot see it on the histology image right now. This is showing you the filtration in the yellow balloon with blue arrows as the molecules are exiting the blood, traveling through the podocytes into the lumen of the glomerular or Bowman's capsule. The same thing in electron microscopy, labeled utterly on the next slide. So this structure is the glomerular filtration barrier or filtration membrane. Barrier between what? The blood and the urine. Okay, so on the left side is the cardiovascular system. This is where the blood is in the glomerulocapillary bed. And on the right side is the urine, where the filtrate or the first urine is sitting in the lumen of the glomerulocapsule. What are the three histological layers that the molecules will go through? From the blood into the urine, represented by the filtration blue arrow. So on the blood side or cardiovascular side, this is the fenestrated simple squamous epithelium. Then comes a fused basement membrane. And then on the urinary system side, these are the filtration slits between the feet of the podocytes labeled with P2. So the green arrow shows you the filtration slits, and these little chunks are the sections of the P2 small podocytes feet. Between the feet, there are some little diaphragms, bridges. So now you can see how the filtration goes from fenestra, basement membrane, and filtration slit. On the right side, this is a three-dimensional electron microscopic image of the podocytes. You can see the large feet labeled with P1. There's another large feet here. The large feet branchy into little feet labeled with P2. And between the feet, little P2 feet, those spaces you can see are the filtration slits. Where is the capillary? Under this. This is the respiratory membrane barrier. This is the filtration membrane barrier. Barrier between what? This is, you remember, this is in the lungs, at the alveolus, the blood and the air. Here is the blood and the urine. Epithelium 1, fused basement membrane, epithelium 2. So this is the three layers of the respiratory membrane: endothelium, fused basement membrane, pneumocyte 1, where the oxygen and the CO2 can go across. On the right side, if you look at it, is very similar. However, the filtration membrane barrier is much more leaky because the simple squamous epithelium is fenestrated. On the blood side, more leaky. And on the right side, the podocytes also have spaces between the feet, much more leaky than the pneumocyte number 1. Reminder, guys, one thing, that the blood and the air never mixing in a normal person. If they do, the barrier broke down and the person has a disease. The same thing in the kidney. The blood and the urine never mixes in the healthy individual. If for some reason this barrier breaks down, we have a very serious disease, kidney disease. Okay, let's look at the blood supply of the renal corpuscle. So we're gonna focus now on the cardiovascular side. And here we go. Number 1 is the afferent arteriole, represented with a larger diameter. This is branchy and enters the renal corpuscle and becomes glomerulocapillary bed. You can see that lies in the pink erythrocytes. This is the first abnormal red capillary bed. Then this is gonna merge, and it becomes an efferent arteriole. Let's call number 3 efferent arteriole. The diameter is smaller. And guess what this guy is going to do? Something very strange. It's going to start branching again, creating a second capillary bed, which is going to be the normal red and blue capillary bed. And again, this is a very unique situation. So anywhere, and you cannot really see the branching on the histology slide, but you can see very nicely on the model. So afferent arteriole, bigger diameter. Branchy, glomerulocapillary. Mergy, efferent arteriole with smaller diameter. And here's the unique thing. Branchy to create the second capillary bed of the kidney. That we're gonna call peritubular capillary, number 4. But on the slides, anywhere you see red blood cells, all around in the cortex, it is the peritubular capillaries in the cortex. The next video is gonna talk about the juxtaglomerular apparatus.